This lecture is referred as integrated metabolism. By right, this lecture should integrate every pathway that you have learned for this biochemistry course. However, there is still certain limitation where lectures such as photosynthesis and even some parts of nucleic acid metabolism is not really covered in this lecture, since the lecture is really relevant to human body rather than covering also plants. In terms of what will be covered this lecture, it is going to be talking about, assume like a person called Bob. He has conditions, the metabolic conditions, such as being fasting for a total of 17 hours. So what happens in his body related to the glucose level, whether it will drop and what effort need to be done to bring it up again. So that's fasting for overnight. Then after that, Bob will go for post-absorptive condition, whereby he eats after 17 hours of fasting. And now what happens to his metabolic states also will be covered in this lecture. Upon these two conditions, we also look into Bob exercising, whereby a lot of adaptation required to be done in his body to achieve maximum energy, obtaining maximum energy. Later, we also look into Bob under extreme condition, such as that of starvation. And this starvation, unlike fasting, is not a matter of one day, but rather up to 40 days and beyond. We'll also look into what happens if Bob gets into a disease state, and the selected disease is diabetes. So all this will be covered in this lecture. So before we look into all the condition that Bob is going to be exposed to, it's best we also look into the vital organs in Bob that will be playing roles as receivers as well as givers. In terms of receivers first, you have the brain that need to be given glucose at all times. The glucose need to have the minimum amount of glucose, or else it will be having a situation called coma, and then also could lead to death. So brain only utilizes glucose. It does not use fatty acid. Therefore, there is no storage of fatty acid. In fact, there is also no storage of glycogen. So it uses glucose from the bloodstream and is not influenced by insulin, the hormone insulin. It can take up glucose without the influence of insulin. Another thing is the glucose is oxidized in aerobic rather than anaerobic, because if it's anaerobic, then it could lead to acidosis, the lactic acid, and it could damage the brain. The only possibility of it resorting other than glucose up to 50% is also using ketone bodies. Ketone bodies comes about from the pathway ketogenesis, where fatty acid oxidation, when fatty acid oxidation such as beta oxidation takes place, it will lead to a lot of acetyl coenzyme A, and acetyl coenzyme A will be so much that it cannot enter the citric acid cycle. It accumulates and causes ketogenesis to be initiated instead, and therefore ketone bodies will be produced, and therefore ketone bodies can then be used by brain. Only ketone bodies used by the brain rather than fatty acid. So that's the brain as the receiver. Then you have muscle. muscle cells, myocytes, they are also receivers where they use glycogen from where it is stored there itself. Glycogen become glucose, and it can be then oxidized in the usual manner, aerobic or anaerobic. When glycogen depletes, it also can use fatty acid, which is supplied by fat cells. Need to mobilize it to the muscle first. It also can use this phosphocreatinine. We later look into what's the meaning of phosphocreatinine. So that's the receiver. The glycogen, which is found in muscle, cannot be converted to glucose and enter the bloodstream. The glycogen is only meant for muscle usage. Next, we have the providers. We have the diet where you consume food. Food could be in the form of carbohydrate, protein, lipid. Then they can be digested to their monomers, sent to the liver, as well as other parts of the body. But in the liver, they can be kept as glycogen and even fat in the form of triglyceride, as and when required through the bloodstream. As you can see here, liver can break down glycogen to up to glucose, and glucose can enter the bloodstream and elevate the blood glucose level. And once it reaches the muscle cell, due to insulin secreted from pancreas, the glucose can then enter the muscle cells for its catabolism requirement. So liver is a good provider to ensure the blood glucose level is always at uh, uh, an amount that is considered normal. So now we look at the first condition that Bob is in, whereby Bob has been fasting for 17 hours since 6 p.m. in the previous evening, and after 17 hours he starts to eat before he goes for the biochemistry lecture. So he has not only undergone the first condition, he also undergo the second condition, whereby in the first condition he is to be fasting for 17 hours. Okay. And then in his second condition, he will undergo post-absorptive or fat state, where he will now have food, and therefore there will be changes in metabolic in both of these conditions. In the first condition, there will be definite drop in glucose, while in the second condition, there will be excess of glucose. In the first condition, when there is a drop of glucose, something need to be done in order to elevate the glucose back to what they refer as a steady state. Okay? So this steady state is values that are considered normal glucose level. So when he is fasting, the level of glucose drops. Then something need to be done in terms of the metabolic pathways to elevate the blood glucose level to not to a single particular value, say 5. It's always 4 to 6. It could be 5.5, it could be 4.5, but it will not go below 4. So that is called steady state, where you cannot get the exact value such as say 5.2 all the time. The body will always go in between this normal range. It could be 4 to 6. But if it goes below 4, below say 2, then it's quite bad for the body, where brain cannot get glucose and it could lead to death. So it must be careful. It always need to elevate back to this steady state. And if it goes beyond say 10, it's also bad, because that means there is a disease state, that is diabetes. Therefore it's also bad for the body. So in order to make sure it stays in this normal range, that's where these two hormones comes in, that is insulin and glucagon. When its glucose is high, it will be brought down by the effect of insulin. When it's low, glucose is low, glucagon will then be secreted in order to raise up the glucose. So this is how it is done to maintain the steady state. So throughout this lecture, you will have this diagram to help you understand in terms of the contribution of the provider, the liver, and the receivers, that is the brain as well as the muscle. You also have the other provider, which did not talk about in the first lecture, first slide, that is the adipose tissue. This adipose tissue also is a provider where it is kept with triglyceride, and that will release the triglyceride by being transported by lipoprotein mostly to that of muscle as well as the liver for beta oxidation to take place. So when it comes to Bob, what happens is when he is fasting for 17 hours, as some of you being Muslim do fasting in the Ramadan month, which makes your body to have low glucose level. So what happens in your body in order to elevate back the glucose level back to normal range? It will have the providers such as liver to undergo glycogenolysis. In the liver alone, glycogenolysis will come to the end product of glucose, not glucose 6-phosphate. In the muscle, it will be up to glucose 6-phosphate, and this phosphate having negative charges cannot bypass the lipid bilayer. But once in liver, it become glucose, it can bypass the cell membrane and come out into the blood, and therefore it provides until elevate the blood glucose level, hopefully back to normal. Apart from that, liver can also do gluconeogenesis. If you recall, gluconeogenesis can take place in the liver 80%, while 20% in the kidney. So gluconeogenesis is a way of utilizing 2 carbon, 3 carbon, such as lactic acid, pyruvate. All these can be converted to glucose and given to the blood system to elevate the blood glucose level. Apart from that, also can use fatty acid in order to be alternative fuel molecule other than glucose. So let's look into the hormones when it comes to fasting state in Bob's body. So during the fasting state, we look at the insulin and glucagon. We have the glucagon. Whenever blood glucose level drops, glucagon will be secreted more compared to insulin. When glucagon is raised up, it causes simulation or stimulate glycogenolysis. So it can cause glycogenolysis by activating glycogen phosphorylase. So I hope you recall the glycogen metabolic pathway, where glycogen phosphorylase is the most important or crucial enzyme in glycogenolysis. It also can cause activation of gluconeogenesis, where can make glucose by using 2 carbon and 3 carbon intermediates. And it looks like it also cause lipolysis. If glucose is not sufficiently made through glycogenolysis in the liver and muscle, gluconeogenesis in liver, then can also resort to use lipid through beta oxidation. You also have a drop in insulin, and this indirectly will encourage lipolysis. In contrast, after Bob has eaten, after undergoing fasting 17 hours, now he eats, therefore food gets digested and lots of glucose gets into the blood and will raise up the blood glucose level. So all efforts needs to be done in order to keep this glucose. Unfortunately, our body cannot secrete the excess glucose through the urine. It's not possible unless you are diabetes. Then blood glucose goes through the kidney and to the urine. But when you're normal, all blood glucose will be kept, and it will be kept in this manner where the glucose will go through the muscle cell, and it will be converted to glycogen through glycogenesis. Remember glycogenesis pathway, where glycogen synthase is the crucial or rate limiting enzyme, and therefore glycogen synthase will be activated during fat state to become glycogen, and glycogen will be stored. Of course, some glucose will be used for glycolysis to run the ATP production for the muscle. Also, blood glucose sufficiently taken for the brain, therefore there will be no problems with the brain energy. And excess of glucose also goes into liver and undergoes glycogenesis, and there glycogen can be stored. Also, if you recall the lipogenesis, that means fatty acid synthesis, once glucose is been converted to acetyl coenzyme A, the acetyl coenzyme A can be utilized in making fatty acid through lipogenesis. So through lipogenesis, you will get lots of fatty acid, and this fatty acid can be converted to triacylglyceride and mobilized to the fat cell and kept as triacylglyceride as and when required. So that's the effect in order to make sure the blood glucose level, which has been raised during fat state, to come down to the steady state. So here again, given the situation on the hormones itself after the state of Bob uh, eating. So when it comes to the hormones after eating, definitely blood glucose is raised up, and what happens is the insulin now will be secreted more, while glucagon will be secreted less. So what happens at the end when insulin is raised up? It will cause all the metabolic pathway relevant to fat storage, glucose becoming glycogen will be activated. So insulin will cause uptake of glucose into muscle cells, liver, and therefore glycogen will be synthesized to be kept there. Also, insulin being a feel-good hormone, when times are good, plenty of food, it also will cause fatty acid synthesis. Fatty acid will be made to bind to glycerol to become triacylglyceride, and then also even the production of cholesterol takes place, and also protein synthesis. All these causes growth also to take place when insulin is present. Here I provide the YouTube video on how to measure blood glucose level using a device referred as a glucometer. This glucometer functions through the theory of biosensor, which you can take up as an elective course in year 3 or 4 of this biotech course itself. In terms of the YouTube on glucometer, you can also watch other that is relevant to glucometer, but I chosen this. Then you have the table there. When you measure the glucose level, if your body has 4 to 6 glucose of millimole per liter, then it's considered normal, no problem. If it goes below to below than 2.8 and below 2, then that's where the coma comes about, or the person just faints, and hopefully no coma. But if it goes higher than say 10, you can see there, that means the person has a condition and it's likely because of diabetes. We move to the next condition of Bob, where Bob decides to do some exercise. So when Bob exercises, he will demand a lot of energy usage because more than one single movement. In fact, it needs a coordination by the brain to signal for various movement. He's not doing repeated repetition of the same movement. Sometimes even different movement required to be done, and therefore brain need to assist. So exercise requires lots of energy. So energy can come about from glycogen, from liver and muscle, fatty acid, and fatty acid using so much will also cause ketone bodies to be utilized. When it comes to glucose oxidation, also possible to utilize aerobic, anaerobic, and also something referred here as phosphocreatinine. You can't keep ATP, but it's possible for you to convert it to phosphocreatinine, and phosphocreatinine, whenever required, can give back ATP again. So that's we'll be covering in a while. This exercise state that Bob is in also requires up to the level of using one more hormone, that is adrenaline or epinephrine, where the fight or flight hormone is also to be playing a role other than glucagon. So above of this slide, you get to see the phosphocreatinine being created from the protein creatinine with the assistance of creatinine kinase. So in the muscle, ATP by hydrolysis will give its energy in the reaction to creatinine with phosphate to become phosphocreatinine. On the reverse, when required, ADP will be reconnected with inorganic phosphate to become ATP by using the energy released from the breakdown of phosphocreatinine. So then ATP can be hydrolyzed for Ng again. So no need to basically make ATP by substrate level phosphorylation or by the electron transport chain. You can use it by what's been stored in phosphocreatinine interactions. So that's on the resources of ATP from creatinine. Then we look at the diagram below on what's happening in the muscle. So now you assume Bob is jogging in the park. So don't look at what's on the asterisk, that is the lactate production and phosphocreatinine. That comes about when there is a burst of activity required. Just look at the others on when he is just jogging like normal. Okay, so when he's jogging like normal, he's using glucose, free glucose, aerobic, become ATP 36, 38 ATP. Then he also uses fatty acid, burn fat that is stored in his body, and a lot of usages of fatty acid eventually will be also cause ketogenesis pathway to be triggered because acetyl-CoA accumulates. You also have when free glucose is finished, can also use muscle glycogen to become glucose, and that also can be aerobically catabolized. And then suddenly a dog comes. Dog chases Bob. So a burst of activity needed where he needs to run away from the dog before the dog can bite him. And that's where anaerobic pathway takes place, where he rapidly uses or rapidly makes ATP where the supply of oxygen is not sufficient to go into aerobic, where demand is much more for ATP rather than supply of oxygen from the lungs to the muscle cell. Okay, so apart from that, bursts of activity also assisted by phosphocreatinine becoming creatinine, and therefore ATP can be produced, and hydrolysis of ATP gives rise to energy for Bob to run away from the dog. So these are uh, effects done in the muscle during, you can put it as exercise condition for Bob as well. This is just a diagram to show the energy ranking of compounds. As you can see here, what we just learned just now, phosphocreatinine, it's in higher ranking than ATP. So phosphocreatinine, once it's broken down to phosphate as well as creatinine, the high energy phosphate can be utilized in making ATP, and ATP in return can be used to be coupled to any endogonic reactions, such as that of glycolytic pathway, where glucose can be converted to glucose 6-phosphate and require ATP hydrolysis. So it's just to show that what is highly ranked in terms of energy, and what is ranked lower, and in the middle ATP. So ATP is a good coupler for reactions from exergonic to endogonic. Here we look at exercise where we can break it down to anaerobic exercise as well as aerobic exercise. But in general, there's no such thing as anaerobic and aerobic. Whenever a person is doing exercise, it's the body shift from aerobic as well as anaerobic pathway. That one cannot be just isolated to be aerobic or anaerobic. But to generalize, anaerobic exercise is like, you run the 100-meter dash in 15 seconds. It's short period, and it will come to a point where because of the competitive nature, you want to go to the push your body up to the supply of oxygen cannot meet the demand of energy, and therefore likely to become anaerobic pathway, where oxygen is not used, and lactic acid is in turn is made. So that's anaerobic exercise. When it comes to aerobic, it's a longer period, like you're running marathon, where you run, if it's you feel like getting tired, you just can walk, and therefore it's very unlikely to go to anaerobic, and that's the meaning here. So this is aerobic as well as anaerobic. So here shown the diagram has been described for the other condition, same here. So when it comes to aerobic exercise, but if you look at it carefully in the muscle cell, it doesn't only give aerobic fermentation or aerobic glycolysis. It also actually goes into anaerobic fermentation, that is anaerobic glycolysis. So in actual sense, there's no such thing as aerobic exercise, where actually the body will alternate between aerobic as well as anaerobic. So also shown in the diagram is the ability of the brain to provide directions to the muscle in order to also absorb calcium. Calcium ion is actually absorbed because of the hormone calmodulin. So calcium is absorbed in the muscle, and calcium facilitate the contraction of muscle. So in order to do contraction of muscle, needs lots of ATP energy, and therefore need to provide glucose as well as fatty acid, where can be oxidized to provide lots of ATP for contraction, and therefore muscle contraction lead to better exercise. So now we look at another condition of Bob, where Bob is being starved for not for 1 day or 2 day, but actually beyond 40 days. In general, human body can survive up to 40 to 60 days without any food, but must have water. Without water, the body cannot survive even beyond 2 or 3 days. So, assuming water is given, then Bob can survive up to 60 days, depending on his own strength and also the storage that fuel molecule that he has in his body. So, in order to survive for long duration, the body will undergo some kind of adaptation. This adaptation takes a duration of 3 weeks or around 20 to 21 days uh, after the process of starvation has been started. So assuming Bob is given water, no problem with water, and then he is left to starve without any food, what happens in his body is there's an early phase, mid phase, and then fully adapted. The early phase is actually the post-absorptive phase where all the free glucose, all the glycogen is depleted, finished, used up for to get energy. Then also his body will start gluconeogenesis, where it can make glucose by using uh, amino acid from muscle, but not vigorously, not extensively until muscle wasting takes place. Just sufficiently and controlled manner. Okay, so from protein can get glucose, and glucose can be oxidized. Then in the mid phase, also start to use fatty acid. And so much of fatty acid beta oxidation takes place, a lot of acetyl coenzyme will accumulate, citric acid cycle cannot use it so much, and therefore shunted towards ketogenesis. This ketogenesis you don't get to learn in this course, but ketogenesis can create three ketone bodies, that is beta hydroxybutyrate, you have acetoacetate, and also acetone. Acetone, of course, it just comes out of your lung. It cannot be used, but the two other ketone bodies can be utilized for energy production. And then after 3 weeks, the body will already now start to use only this gluconeogenesis, fatty acid, ketone bodies, and survives as long as possible. And then if still no food given, then problems start to occur where it start to use muscle proteins to the extent where muscle wasting takes place, and that's where the danger start to lie. So here shown in terms of what happens is Bob's body in terms of the glucose being used in his body and also the ketone body production. Okay, so you have phases 1, 2, 3, 4, 5 being shown there, or stages of 1, 2, 3, 4, 5 stages. So initially in the stage 1, hope you can see there is a line there, very faint, but that is the line of glucose, where the free glucose found in the blood is used, post-absorptive glucose, where you can see within 4 hours all finish already. Okay, and then the muscle as well as liver start to use the glycogen through glycogenolysis. So, but that also within 28 hours completely finished. What happened next is the gluconeogenesis start to take place, and initially it's can achieve production of lots of glucose because there's a lot of intermediates like lactic acid, pyruvate, propanic acid, and also certain amino acid that can enter the gluconeogenesis. But after that, since there's no food given, even that start to drop, and within like 2 to 4 days, you can see it's dropping and then kept to plateauing amount throughout up to the 40 days. Okay. So after this gluconeogenesis, the body need to rely on fat, and that's where rigorous use of fatty acid by beta oxidation will lead to ketogenesis in turn to be activated, and ketone bodies are now produced. So the brain will have a drop in glucose usage and rely on ketone body as well. Up to even 50% can use ketone body for the brain. But beyond 40 days, by using glucose from gluconeogenesis and also fatty acid and ketone bodies, if still no food been provided to the person, that's where the body start to use the muscle protein, and muscle wasting takes place where muscle protein converted to amino acid through gluconeogenesis can sustain glucose. But when the muscle is gone for a person, the person will become very weak, and over time death could take place. So here summarize in terms of what's happening throughout the journey of adapting in the condition of starvation, that is 2 days to 3 weeks. So you can see between 2 days to 3 weeks, the body start to adapt. How does it adapt? It does ketogenesis, increases where glucose drops, therefore can rely on ketone bodies instead for the brain. And then gluconeogenesis will be stimulated at the maximum level, where it can use these 2-carbon, 3-carbon precursors such as that of lactate. Lipolysis, beta oxidation also can be activated to be happening at maximum, and lipolysis uses fatty acid. Fatty acid comes about from triglyceride. When fatty acid taken from triglyceride, you also have glycerol, and glycerol also can be precursor for gluconeogenesis. And then you also have the person becoming weaker and weaker, not as active like normal at normal time. So they will be much more docile rather than active. And lastly, the ketone bodies, at least for 2 to 3 weeks in the adaptation period, it can spare the muscle protein. But beyond that, coming close to 40 days, it is likely muscle protein in turn can be utilized for the purpose of gluconeogenesis, and that's where the danger takes place for the person. So again, we come to this diagram which shows the what happens in the condition of starvation after adaptation to the state of starvation. And you can see that the body using whatever resources in order to get fuel molecules and up to the level of ketone bodies itself. Okay, if ketone bodies is not working, you will have a situation where brain does not have enough glucose, and that will be very much detrimental to the body. You also can see the resources of fuel molecule is to the level of using muscle protein, and that means degradation of muscle protein will cause a huge problem to the body itself. So all resources uh, are used in the times of starvation. So now we talk about Bob under disease state instead. Um, Everybody experiencing any disease, even the common cold, will be exerted in terms of energy production. In the normal condition without disease, you will have energy for normal functioning of the body. But when you have a disease such as the common cold, the body needs energy for another purpose, that is to build the mechanism to get rid of this infection. Uh, the infection is removed by using energy to build antibody, for instance, and that requires lots of energy as well. So much so, you are made into become weak and to just lie down to overcome this infection. So that's how serious it could be. Uh, when it comes to looking at the metabolic diseases, can look into lipid diseases such as that we already learned in the lecture for lipid metabolism. If you recall the carnitine, people who undergo hemodialysis or preterm babies may have issues where the carnitine amount is lower than the normal amount, and carnitine is very important to bring fatty acid from cytoplasma into the mitochondria. That means cannot do beta oxidation using fatty acid and have to rely on carbohydrate. But it can be overcome by just giving carnitine to these individuals, such as a supplement, and hopefully that can overcome the shortage of carnitine. So that's lipid diseases. But we are selecting diabetes to look into in detail here so that you understand when Bob is in this extreme state of disease. So before we look into the condition of diabetes, it's best to look into the function of this very important hormone, that is insulin. So insulin is normally it's called the feel-good hormone because when conditions are so good for the person, as food is given in more than excess, so it will cause the digestion of carbohydrate to become lots of glucose, and in the bloodstream it will accumulate, say outside of the muscle cell. And insulin will be triggered from the beta cells of pancreas, and it will then come to the insulin receptor at the muscle. It will bind and cause the activation of IRS, insulin receptor substrate. When IRS is activated, it can cause simulation of various action, and includes that of glucose uptake, where IRS will activate glucose transporters, uh, also known as GLUT, where it will cause the channel to open, and therefore glucose can come in from bloodstream into the muscle, and that will lead to glycogenesis to be activated, where insulin will cause uh, dephosphorylation of glycogen synthase, and therefore glycogenesis pathway will be activated. So glycogen storage can take place. Other than that, could also cause fatty acid synthesis, that is acetyl-CoA carboxylase to be activated, and then fatty acids can be made. Palmitic acid, you want extension to become stearic acid, you want to put uh, double bond become oleic acid. All this can be done and then made into triacylglyceride. Insulin also encourages cholesterol synthesis and therefore cell membrane growth all can come about. How it activates growth or replication is to activate initiation factors in the cell. And therefore, transcription, translation can take effect. One example I can give you is the making of fatty acid synthase. Remember that lecture where insulin encourage fatty acid synthase to be made through transcription to translation, and therefore a lot of fatty acid synthase is then found to make into fatty acid. Indirectly, insulin also inhibits lipolysis because if you already have lots of glucose molecule, there is no need for you to rely on fatty acid. So this is the IRS, which can call is a signal transduction, where one of it is actually allow glucose to come in from outside of the bloodstream into the muscle cell to be made into glycogen. So this is the diabetes, where it can be divided into two types of diabetes, that is type 1 as well as type 2. The type 1 is also known as juvenile diabetes, where it can the person is having a defective gene and they are actually born with the condition of diabetes. The outcome of this juvenile diabetes is the person does not have any insulin secreted whatsoever in their body, and therefore it will cause elevated glucose in their body, and they have to inject themselves with insulin, provided insulin from outside of the body by injection throughout their life before every meal. So if they do not inject or miss an injection of insulin, it will cause usage of fatty acid to an extent where ketone body production will escalate, and that uh, ketone body production will in turn cause ketoacidosis and therefore numerous other complications in the person. The type 2 diabetes is due to non-insulin dependent, and therefore they do not have to rely on insulin. So no need to inject insulin. This one normally happens to people who are say 40 above or 50 above, more likely in this group of people, because they become obese and therefore not enough insulin, or insulin is secreted but it is resistant to doing its function. So still high blood glucose, but some glucose do go into the cell, and therefore still can rely on glucose rather than fatty acid. So it's not to the level of relying on fatty acid solely until leading to ketogenesis. So here I compare using this diagram again in terms of the condition, that is untreated type 2 diabetes mellitus. We take that first, where you can see blood glucose will be high, and for type 2 at least there are some insulin can still do its function, and therefore some glucose can still go into the muscle cell, so much so that it will not cause fatty acid to be utilized at all time in a vigorous manner until ketone body is produced. So the fatty acid in type 2 will not be until the effect of using ketone bodies. But this is not the case for type 1, where there is no glucose capable of entering in the muscle because there is no insulin in the first place. Therefore, relying more on fatty acid until lots of acetyl-CoA accumulates and no choice ketogenesis can also be triggered and ketone bodies will be generated, and this will lead to possible of ketoacidosis if not treated. Of course, the meaning of untreated for both type 1 as well as type 2 is also possible. The person given drugs or injected with insulin, then the story is different here. The diagram here will be different, where the blood glucose can actually go into the muscle cell and there will be no requirement of ketone body to be generated, and also in the type 2 there will be unlikely of using so much of fatty acid. So that's the difference. So that concludes the lecture of integrated metabolism. Here there are a number of questions for you to look into when you study for this lecture itself. Thank you.